ask some other questions. I would like to ask you to just give a brief detail of your current research topic you are working on right now.
Right now, I'm a postdoctoral fellow with Tamal, Das's lab at TIFR Hyderabad. So I am working on patterning of hair follicles. So if you have ever noticed, like the hairs on your hand or any part of your body, they have a uniform
Mhm.
distance between each other, right? So how
Yeah.
do biologically that regular distance is generated molecularly? Well, I'm trying to study that. I'm using mouse embryonic system, and we look at a
Mhm.
stage of development where hair follicles have not appeared. And then we
Okay.
take out a skin patch and see how hair follicles are now appearing and see what could be the reason behind placing them at such regular distance. So hair follicle patterning in mouse embryonic skin. It's unpublished
Whoa.
right now. It's just
That,
exploring something. Let's see where we go.
yeah, sounds interesting. So, like, you have to work in wet lab as well, or you just
Yeah,
develop
mostly wet lab.
embryos and okay.
Mostly wet lab. We get pregnant mice around
Mhm.
12.5 to 13.5 of embryonic development, and then we use
Okay.
them.
Okay. And everything you do in the TIFR only, right? Tata Institute of.
Yeah, yeah, it's in Tamal's
Mhm.
lab. Yeah, right now in TIFR. Yeah, yeah.
Okay. Okay. So now you're like properly as a scientific work you're doing. So you wanted to become scientist from the time like when you were in,
Yeah,
I think your schooling was from, I was just saying Jawahar
yeah.
Navodaya Vidyalaya.
My schooling was from Jawahar Navodaya Vidyalaya, but taking up this academic career that happened when I during my BSc MSc I think like I, after
Okay.
class 12th I think my parents wanted me to take engineering or medical and I did drop 1 year to do that and then I was like bored of it,
Mhm.
so I just joined BSc and at that point I knew after
Mhm.
BSc MSc PhD you get into research, so I liked it. I thought why not, I really like doing science, so yeah.
Mhm. Okay. Before I get to the another one, one question you have got for a lay audience not familiar with the terminology, could you briefly talk about your work?
I'll talk about my PhD work, not this
Mhm.
other postdoctoral work. It's just starting, so not much there to talk about. I'll talk about my PhD work. So I was working
Okay.
on wound healing. So if you
Mhm.
get a cut on your skin or something, it always heals, right? It all the gap always closes. So I was working on wound
Mhm.
healing. So we're
Mhm.
using a cell line model to study wound healing. So during wound healing, let's say
Mhm.
if on your skin you get a cut, the skin is open and the internal part of your body is exposed to the environment and germs or anything they land in, you will get
Mhm.
infection. So to do that, the skin cells, also known as epithelium, they close the gap as soon as possible. To close that, they need to move or migrate towards each other. There is cell like locomotion, they move towards each other. So people have been studying many aspects of it. I was studying one particular organelle that kind of gives the cell a direction. Is it top or bottom or is it front or back? I was studying that organelle. It's called Golgi apparatus. It's generally associated with protein secretion and sorting, but in cell migration, it plays an important role in defining the cell direction. Is it top bottom or is it front back? Because you need to
Mhm.
migrate towards the front side to close the wound. So Golgi is
Mhm.
important there.
Okay. And
So in my PhD, I found that if Golgi doesn't go through a certain stage of this certain changes, the migration is not directional. So you need your all the cells to migrate in a particular direction to close the gap. It cannot be random, right? So
yeah,
Golgi has to go through a certain change that allows the cell to migrate directionally.
okay.
I hope that
I can
was
see that.
layman enough.
Yeah, yeah. I could really feel that the terminologies were coming and you were trying not to. So
Yeah,
those
yeah, I'm trying.
about the polarity of Golgi bodies and other
Yeah,
things.
yeah, 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 yeah. Actually,
So
I was fascinated with
actually,
Golgi during BSc. So during BSc, you
okay.
must have also read in Cooper's cell biology, we read
Yeah,
about cell sorting, right? Golgi protein sorting to different
yeah,
compartments and plasma membranes. I was really fascinated by
yes,
that
yes.
process. And
Okay.
coincidentally, for my PhD, I came across a project where I had to work with Golgi, so I was really happy about that.
Mhm. Yeah, in fact, I was coming to that question, like why specifically Golgi bodies or Golgi apparatus? And you have just answered because you were fascinated during your bachelor's itself. Uh, but
So
before that, yeah. Please continue. Yeah.
So the PhD landing on Golgi during PhD was kind of a coincidence. I was looking at wound healing. So
Mm-hmm.
people just in cell culture system, people just let cells migrate towards each other and look at different internal part of the cell and how they behave. I was looking at this protein called E-cadherin. It joins cell to cell to like two cells
Mm-hmm.
contact each other through E-cadherin. And what I found, E-cadherin should be at cell junctions, right? If it is a contact protein. But I
Yes.
also found a big cluster near nucleus, which I guess probably was Golgi. And that cluster didn't appear
Mm-hmm.
homogeneous. Not always Golgis are clustered, but they had a very unique organization. It's called, I call it an equatorial ring. You can imagine like the Saturn ring. Golgi was taking up a position like that around nucleus. So that was something new I found, and then I read literature like nobody has seen this. People don't know how Golgi moves during migration. So I spoke to Tamal, and Tamal was like, "Yeah, nobody has looked at it. This importance of Golgi for migration is known for about 50 years, but how it happens was
Mm-hmm.
not known. So after speaking to Tamal, we were like, "Yeah, this sounds fascinating. We could pursue it, and this is how it happened." Yeah.
Okay. And oh, nice. I was actually thinking about that only. How you landed up specifically to the Golgi. And somewhere I read your article that it is some 50 for 50 years or so that it was like an enigma to solve. And you,
Yeah. So So you must have heard of Camillo Golgi. So he used to do
yeah.
silver staining for tissues, right? And he had stained for many Golgi Golgi organization,
Mhm.
different type of cells. So
Okay.
one of his tissues he had looked at was lactating mice, and during
Okay.
different stages of lactation, Golgi used to go through different kind of organization change. So something like
Mhm.
that was known, but this equatorial position was not known. People always found that whichever direction cell migrates to, Golgi gets reoriented, Golgi also
Mhm.
pointing towards that direction. But how that is maintained, molecularly, how a cell is putting that Golgi towards a
Mhm.
specific direction, that was not known. And in context of
Mhm.
migration, that is what I try to
Mhm.
look at.
Okay. And now as my field has completely changed to the taxonomic and other fields.
Yeah,
Now
yeah,
if we talk if we talk in this sense that it moves in from this direction to this direction, someone would imagine that as you are telling, then you saw it moving this direction and that direction. One can get this impression that we might be seeing under it like microscope or something, and we can
We
see that
often.
3D things going on and live we can see like a video. How you actually did this research and you found out now it is moving. How it actually you found
Yeah.
out.
So first, when I saw that E-cadherin thing, we just stain the sample and use a fluorescence microscope. But with a better microscope like confocal, which has a better resolution in XY, so you can resolve the distance between two units smaller and smaller. There, if this is the entire height of the cell, and if you take small section in between, you can make out in the
Mhm.
3D organization of the cell where are different organelles. And my interest was Golgi. And when I looked at my samples, I could see three different kind of organization. If this is the entire 3D volume of the cell,
Mhm.
in some cells it
Mhm.
was at the top, in some cells it was
Mhm.
towards one side, and in some cells it
Mhm.
was taking a equatorial ring like the Saturn ring I was talking about. So yeah, you can look at and pick samples in 3D, but to see
Mhm.
live, I actually put a protein of my interest which has a fluorophore tag to Golgi
Mhm.
membranes, and with time I can track how Golgi is moving
Okay.
in
So it was a
fluorescence or
continuous.
confocal microscope. It's a continuous process, yeah.
Okay. And cells won't die in between.
No, so we give a condition where cells are like happy to grow, like we maintain the temperature and pH. Under the
Hmm.
microscope, with the light we are using, laser we are using, it could induce some phototoxicity, but we kind of try to stay in a range where you can still see, but cells are not dying. Very low power of laser.
Okay. And we will be very happy to know which kind of cells you are talking about that you worked on.
I was using a cell line called NDCK. It's a epithelial cell line derived from dog kidney cells. The kidney
Okay.
epithelium, it's derived from that. It's an immortal cell line, MDCK.
Okay. And one may can simply ask, like as I'm in this field, why not directly to the human cells?
Working with human cells, you need a lot of ethical clearance. And
Yeah, yeah.
if I talk about feasibility problems, so getting reagents for non-human cells, they're first less
Mhm.
expensive and easier to procure. For human cell lines, you will need ethical clearance and all the specific reagents for human cell line. What I came
Mhm.
closest to was using mice intestinal epithelium. So we can get mice,
Okay.
dissect out
Okay.
a small piece of the intestine, and use
Okay.
that to study this migration or wound healing that I have done with this. Same thing, whatever you use with MDCK, I found the same thing with intestinal
Mhm.
epithelium also, mouse intestinal epithelium.
Okay. And these cell lines you were getting from outside or you were preparing?
So
No, right?
MDCK is a commercially available cell line, so you can buy
Okay.
it off from ATCC. But intestinal epithelium, I
Okay.
actually generated from mouse intestine. Like we have a mouse facility here. We just get a mice and
Mhm.
sacrifice the mice and use their intestine. Intestines
Okay.
have stem cells, so they're easier to form a monolayer.
Okay. Okay. Understand. So you'll have to kill one, right?
Yeah, we will have to.
Uh, what was the frustrating point? Sometimes we all go through when we are going through a PhD, then we do go through some phases where we're like, I think this is enough, and sometimes bit frustrated and irritated. So last time when I was talking to one of the guests, Afreen Hussain, so she was like, usually the third year is that point when you are like just stretching your limits. So what was that point in your PhD journey? If it
Uh,
was, if there was.
there was. Yes, I'm not immune to frustration. But
Mm,
for me, experiment wise, I have never felt frustrated if something is not working. For me, like I know there
mhm,
must be a solution which I am not able to see and I'm missing.
mhm,
Frustrating part was actually writing one of my first papers because I am after a point after reading at my whatever I have written the word document, like I get blinded by the words. I don't edit
mhm,
them well. I just dump data. I don't try to construct a sentence that makes sense scientifically. So that
mhm,
was very frustrating because you have to spend some time. You have to have a clarity of mind to the sentence should make scientifically as much sense as it means to you, to the audience also, whoever is reading. So that took
yeah.
some time to learn. So, but when I did it for the first time, it was like my writing was bad. I had to really improve. But now it's
Mhm.
fine. That was the most frustrating part, just editing the same document multiple times. Yeah.
You have got uh, you have got these grants, so definitely that says it has not only just improved, it has improved quite well. So,
Yeah. Actually, but I have written to many labs for postdocs, so that was one exercise I had in my mind. Each time I write to some lab, I'm
mhm,
improving on my writing skills. So I think that did work
mhm.
out with two of these grants. Yeah, let's see.
Yeah,
But there's
we'll
still
come to
more to
that
learn.
grant
I have
because
to keep
that is
working on it.
yes, definitely. We'll come to that grant point because it's an elephant in the room. So we'll definitely. But Uh, now you're like completely inside your research, but I think your first article must have been the one you worked in Bachelor's on
Yeah,
it was
yeah,
some
yeah.
water quality.
Water quality testing. It was the Delhi University project. At that time, I think the university was promoting a little bit of research culture at graduation level, BSc level. So many of my batchmates from biology and some couple of people from chemistry, we were actually checking water quality from different parts of Delhi for biological load and chemical TDS basically. So and yeah, that was published in Due Journal of something, I'm forgetting the name, but that was my first publication. But I had more data contribution. Writing part, I was not. I didn't know at that time.
Okay. And somewhere you have mentioned like your curiosity was very much, driven by when you saw a hibiscus pollen grain going in a glucose solution. Was that
I
in the
actually have
lab?
to check that. Is it glucose or sucrose? I must have. I'm a little confused. It could be glucose or sucrose,
Okay.
but it was the pollen
Okay.
germination. So this was back in I
Okay.
think 11, 12. We had we
Hmm.
had a new biology teacher, and
Hmm.
he was very good at drawing biological. We at the time he used to draw a lot of
Hmm.
biology, right? The structure, the anatomy. He was really good at that, and he had.
Hmm.
For us, he did this pollen germination experiment. So each of us got to get a flower and collect the pollen and put them in that solution. And if you see in your hand, it just appears yellow dust, right? Like smooth to touch,
Hmm.
it just appears yellow dust. But under the microscope, it had really beautiful ridges all over, like spikes at uniform distance. Like
Hmm.
it was something like some viruses have, like the coronavirus or other viruses, they have that spiky structure on the surface all over the 3D sphere. The pollen appeared like that, and it was bright yellow, and it was fascinating. At that point, I didn't know I was going to do science, but when I look back now, I think that was my most memorable moment of thinking about life, how things work under microscope.
Hmm.
Yeah,
Okay. And that was your school time you're talking about,
school
right?
time. Yeah, yeah. Yeah.
So and that was a thing which motivated you to take microbiology for bachelor's?
To yeah, exactly. So in 11, 12, we do study a lot of biology, but microbiology section is pretty less. And because this pollen was so small and in micron scales, I was like, hey, let's study more of this. And I had picked microbiology or physiology or botany or other courses that were offered around that time. Yeah, that was the one of the main motivation.
Okay. And what kept you going for masters in microbiology? Because I hear there are, I think, some 5 or 6 positions in MSc when it comes to Delhi University. You did it from
Yeah,
Delhi University, right?
yeah, yeah. So during BSc, you mostly read about a lot of microbiology and do experiments about seeing different kinds of organism around you. Uh, in MSc, you get to work on those organisms. So it's like during BSc, you get familiar with different kinds of microorganisms that exist, their properties. In MSc, you can pick one and do something with that. There were different labs in DU South Campus, like working on yeast or bacteria or leishmania. So after BSc, after getting familiar with this kind of microbe, I was like, "Okay, why not like work with one and see how going deeper
Mhm.
into one particular organism and see things further deeper into the cell." So I think I'm always zooming in further into the cell is my thing.
Micro world.
Yeah,
That
micro world.
Leishmania reminded me of Srivastava sir's classes.
Oh, yeah.
And and it was during your masters you did one summer training sort of training in CCMB, Hyderabad.
Yeah. You know all the details.
So
Yeah,
there you
I
worked on
went.
some overexpression of.
Yeah, so there I worked on a plasmid where we were trying to
Mhm.
create a plasmid to overexpress a specific protein in Leishmania. And that was to study host
Oh.
immune interaction. So Leishmania with some specific virulence and its
Mhm.
interaction with the immune cells in mice. Yeah. That was a
Okay.
very small part. My role was just to get the plasmid inside the cell, efficiently. Yeah. Yeah.
Okay. Because
But
that's a
that was a
bottleneck
very good
to get.
exposure to, yeah, that is a
Mhm.
bottleneck, and it was a very good exposure to how research labs, other than what I have seen in microbiology, how other
Mhm.
animal cell-based labs work. So they had leishmania also, and as well as mouse cell line work. So it was a good experience. And in CCMB, I also got to know all other different kinds of research labs that are there. Like mostly I was familiar with microbiology till then. After that, I was animal cell culture, plant cell culture. Sometimes it's not even cell culture; it's just an organism,
Mhm.
Drosophila or mice. Yeah,
Yeah, yeah. So because you have been to CCME, which is in Hyderabad, so that's why you decided that you'll come to Tiffer. It's in Hyderabad, or because it will be comparatively near to your Odisha, your
actually,
hometown.
neither of those were more practical
Right.
reasons I joined TFL Hyderabad because I didn't get a PhD in anywhere else. Other maybe
Um,
one more, but I like this one more. Mostly because I didn't get a PhD in any other institute at that time.
okay. And that time you joined Tamal Sir's lab, and you
Yeah, yeah,
started working on Golgi
yeah,
apparatus.
yeah.
Although you like your main work is about Golgi bodies and all, but I was seeing you have worked on Drosophila, gene expression Drosophila. You're related to these kinds of work.
Uh,
Foot and mouth virus infection in vaccinated cattle, then mechanosensitive dynamics of lysosomes. So along with your
I'll come to
PhD, you were doing. Yeah, yeah, please.
yeah. So the lysosome work is actually one of my lab mates in Tamal's lab. So I was helping her with some other experiments related to that work. The foot and mouth disease was my MSc thesis project, which got published really little late. And the Drosophila work was actually one of my lab rotations. So before joining your lab in TFR Hyderabad, we get a option to rotate with different lab and see if you like different kinds of work. I had 3 lab rotations,
Mhm.
and this lab rotation was
Okay.
with Operative Mazumdar from TFR Hyderabad. So in that lab, I was
Mhm.
working with a PhD student, then PhD student Nikita. She was working
Mhm.
on standardizing a specific fluorescence technique in Drosophila tissues, all
Mhm.
tissues, and I was working with the Drosophila embryo at that point. Yeah. So that is from
Okay.
that work here. But my main focus has been Golgi during my PhD, for my
Golgi.
PhD.
Oh, that has been your main focus
Yeah.
throughout. And your PhD lies in COVID time. How was that for
Yes.
your PhD?
Yeah. So that was around fourth year of my PhD, I think, if I recall properly. So
Hmm.
it was a shock, like to everyone else. It was a shock to me also. But
Yeah.
with time, like about 15 or 30 days of complete lockdown. But eventually, in TIFR Hyderabad, we started doing lab work in shifts. So that at a time in lab, there will be only 2 or 3
Mhm.
people and not working very closely. So we used to work half a day.
Mhm.
So if any experiment requires longer incubation, somebody coming in the next shift can help you out. So we were working like that. So that
Okay.
also helped. Instead of no work
Mhm.
at all, there was some work at a little slower pace, slower pace, but that was fine.
Okay. But that did affect your
It
uh,
did slow
obviously that's quite
down.
obvious. Yeah.
Yeah, yeah,
Hmm. Must have slowed down. And then
yeah.
your the postdoc work, what you are doing right now, that has something to do with Golgi bodies?
Uh, no, it doesn't have
No.
anything to do with Golgi yet. Who knows what we will find? But at this point, I'm in this particular work. I'm not looking into the cell as much as cells and higher scale-wise cells or
Mhm.
group of cells. So hair follicles are bigger group of cells, right? So I'm looking at a
Yeah.
dimension where I'm just looking at cell level properties, not intracellular properties.
Okay. So it's
I want to move into a little bit into
comparatively macro.
development. Macro than organelles, but still in micron because I have to use microscopy to study all this. Yeah.
Okay. And now let us come to your this fellowship grant, what you got, Rowland Fellow at this Rowland Institute in Harvard. So I was just seeing you got shortlisted for final interview. There were 10 candidates, and you had online
Yeah.
interview for that. Before that, I was seeing their process, which includes actually 3 4 things, important things, including elevator pitch. That was something new to me to see that. then a statement of research, then CV, and as usual that vision statement, and 3-4 references. Among these four, like first I would like to know about this elevator pitch where you are restricted with 250 words, and
So
how you manage to convey your topic, which is such a technical in terms of the words, to the general audience. How you made that 250 words?
uh, one point here, the elevator pitch was not there during my time. It has been introduced this year. So, but elevator pitch is an important thing to learn because lot of interaction or scientific interaction with other researchers, you don't get enough time to sit down for 1 hour and talk about. You don't get that much time. It's like 5-10 minutes, and you have to convey your interest, and you also have to ignite the other person's interest during whatever you are saying. So I have done it in written format. So I would just suggest talk about the most fundamental thing about your work, and whichever grant people are applying to or whichever whoever they're talking to, how that provide a context that they will also
Mhm.
find your work interesting. Something like that.
Okay.
Not too much
Okay.
detail. If
And
somebody does get interested, they will ask you further. You will get more time.
true, true. And any tips you would like to give for research statement, that 3-page proposal?
So, for specific fellowships, I can. For Rowland, I think they look for. So, this is a fellowship right after PhD. So, they're looking
Mhm.
for really motivated and enthusiastic researchers
Mhm.
who have a little bit of ability to know have a group dynamics, proper group dynamics, so you can work with a team. All of us are going to work
Mhm.
towards one particular goal. So, the research idea, I mean, it should be a good idea. Obviously, somebody is investing so much money, it should be a good idea. And for
Yeah.
Roland, it should plan to achieve a major portion of it within the 5 year span they are providing the fellowship for. And add a little bit what will be the long term vision of that work. If you can ask a question, in 5 years, the research question
Mhm.
would be answered to a certain extent. In 10 years, that research question can become a specific context or niche of a work. So that kind
Mhm.
of thinking, if you have, you can add it to your proposal.
Okay. And
And
how
one
was your
more
interview?
thing,
Yeah. Yeah. True.
just
True.
one
Yeah.
more
Please,
thing.
please.
I
Yeah.
think that is also
Yes.
important. So in the proposal itself, like whoever is reading it, like the role and whoever is checking it, they should be able to see that you have thought about your idea. Like it's like we get ideas all the time, right? Random ideas come, we don't spend time on each idea equally. So for your research proposal, it should be an idea where you have spent enough time thinking, looking at different aspects of it. It can be approached through multiple angles. You have to pick the best angle, which shows you can do it, you have the skills to address that particular aspect, and how it is going to work with other aspect people are looking at in different labs or different fields. So yeah, so you need to spend time on it, and it should reflect in your proposal that you have thought about different aspects and you have picked this one. Yeah, like that. What were you asking? Sorry.
Nice. Yeah. Nice takeaway actually to write proposal for even sometime for PhD also. Sometimes they ask if you are going for
Yeah, yeah.
uh, proposing a research. Yeah. So I was asking as you got shortlisted and you had an interview. Interview must have been good as you have got that now. So are there any generic questions what you would like to share that they asked?
Generic questions, maybe not very generic, other than about running a lab or something, or thinking about the idea from working with a team. Like you doing your own research work for your paper is different. You working on a team for a research question is different. So that could be one generic thing. But since in this interview there are variety of students with different kind of backgrounds, so
Mhm.
and in the interview panel there are different diverse members, so they do get to ask more specific questions about your proposal. So whatever you put in proposal, basically the proposal should be very clear in your mind.
Mhm.
And they did have
Okay.
this thing in a 30-minute or 20-minute presentation, 5 or 10 minutes on your old work, and 10 minutes on your proposed work. I think that was important because if you spend too much time on your past work, people won't get to know what you want to work on in the next 5 years. So stick to this time limit. Don't overspill into your proposed research work. Your proposed plan is more important than at this moment than your old work. So, yeah.
Hmm. Okay. So, and how long it went, your interview?
Everyone, it has to be within a time limit, 30 minutes. 30 minutes for each, and it was actually in two parts. First part was just a presentation and some question and answers.
Hmm.
The next day you had one-on-one interaction with different fellow members. That was for
Oh,
about 30 minutes or 40 minutes with each. Three people I had. Yeah,
oh, oh. And you're going to join there by December this year?
I'm going to join there in December this year, hopefully by first week of December.
Okay, nice. That's great. And along with this, you also got one HFSP, some
HFSP,
long term.
yes.
Yeah.
Long term. It's a
What was that
postdoctoral
about?
fellowship, long term. So before I had this Rowland
Mm-hmm.
thing, I had applied to a couple of labs, and there the idea was to write a grant together to work on the question we all of us in this particular work came up with. So that was
Mm-hmm.
HFSP. It's a very prestigious fellowship again. So with that, I was
Mm-hmm.
planning to work with two different labs.
Mm-hmm.
One lab's expertise, Matthew Rousey. That lab's expertise one was on imaging and 3D, basically highly resolved 3D imaging, and a lot of
Mm-hmm.
quantification from images. And the other lab was
Okay.
Valentina Gigliola, who works on zebrafish. So the idea was to study spinal cord regeneration using zebrafish as a model and trying to address some mechanical aspects of it. So actually, one interesting thing about zebrafish spinal cord regeneration is if you completely cleave it two ends, they still regenerate
Mm-hmm.
and work fully. In mammals like human or mice, the
Oh.
regeneration is not full. So you may recover some locomotion, some activity back of your spine, but it won't be fully as it is in zebrafish laboratory. So that was what we're trying to study.
Oh.
Yeah, so the HFSP proposal was for that work. Since I got the Rowland, I had to say no. HFSP announcements come little later. By the time I had accepted the Rowland, fellowship.
No, this was sounding quite interesting. What is actually you are going to work in Roland one?
Rowland one, I'm going to work on a tissue called pseudostratified epithelium. So if I
Okay.
just break down the scientific terms, pseudostratified, pseudo means something fake, it appears something. So the pseudostratified is a stratified epithelium which just appears as stratified, but it's actually not. And stratification is just layers of cells. If you have one layer, it's not stratified. If you have multiple layers of cell, it's stratified tissue. And if you have one layer of cell, but the nuclei appear in different layers, that is pseudostratified. So I'm going to work on
Okay.
this epithelium and see how packing of nuclei, where nucleus are positioned along the tissue height, affects the tissue properties. Basically, yeah. I want to move
Okay.
into a
And
little bit into
what?
development and organelle to keep my basic interest of organelle oriented research, but to address tissue level properties. Yeah.
Okay. And what will be its the work you are proposing? What are its long-term impacts? Like how it is connected to the maybe a longer some disease or something? Is it related to something like that?
Not disease, but a very long-term plan is to all the developing tissue in our body, and many different kind of organisms, be it Drosophila, mouse or human, they start from this pseudostratified nature. So if an
Mhm.
organ has to develop, it starts from a pseudostratified mass. So I plan to understand nuclear position in such a way that can I keep a pseudostratified epithelium in that state and just provide different cues, growth factors or mechanical, and let them differentiate into different types of organs. Can we generate different types of tissue type material or tissues from the same pseudostratified epithelium? That I see a little bit medical application because many of right now organ regeneration in human is not It's not as efficient.
Mm-hmm.
Other than liver, maybe other organ regeneration is not that efficient. But something called
Mm-hmm.
organoid, which is in between organs and cells, it is physiologically
Mm-hmm.
really close to the organ. So I'm thinking of generating different tissue organoids from one pseudostratified epithelium. So that's a very long-term goal. Let's see.
Oh, that is, and we used to hear actually. I used to see the news transplantation of different
Yeah,
body parts
yeah,
and all. So
yeah,
maybe if this happens, then that will advance that
yeah, yeah. Let's
acceptance
hope.
rate of the body. Mm.
It could. We can take the tissue from the same person or same, let's say,
Mm.
subject of interest and put it back, generating a specific type of tissue. We can put it back in the microenvironment and see. But that's a long-term goal. First, I have to establish that finding that
Yes,
nuclei are
yes.
important and I can control pseudostratification. Let's see.
Nice. So you never thought, you must have thought definitely to apply for this postdoctoral fellowships in India, like Inspire faculty and others.
I actually do want to go abroad for my postdoc. I want to learn few things that some labs outside India, like it's more of their expertise. So there is a person called Karen Norden.
Mhm.
She's one of the pioneering person about pseudostratified epithelial biology. So
Mhm.
I plan to work with her and see what I can learn from them. So her lab is in Portugal. So we'll collaborate, but many of the things that I want to learn or add to my what I already know, my skill set. So I think they're outside, and I do want to see academia and academic life outside India a little bit. Let's see.
Okay.
So I want to, yeah,
Okay. So maybe you might come back after 5 years, 2029.
maybe let's see,
Maybe, yeah. So as you just
let's see.
mentioned about academia and research, I was saying it was I think it's your words only. With single-handedly, you can achieve a lot. You can move a mountain, but with the help of some people, it will be easier. So how helpful was your mentor, and what's your opinion? What's the importance of your mentor in your career?
So that's the first thing I did adapt that line from a different quote. So a little bit I
Yeah.
adapted. It's not mine.
Yes.
I actually believe in that sentence a lot because a supportive guide and a supportive team is very important for you. Because many of the times,
Mhm.
first thing every time with my guide Tamal, like the interaction
Mhm.
was like we discuss science wherever we feel stuck, that we are not able to imagine a solution or think of a direction which to follow. Like he's always there. You just need to talk to him. He will come up with different ideas that
Mhm.
could work, and we will both discuss. If it works out, we'll pick one. So that kind of discussion is very useful because a PI or a guide has many years of extra experience on you, right? So thinking about science or problems that happen around doing a particular project, they're better able to
Mhm.
navigate out of when you're stuck, experiment is not working or which if you have five different leads, which one to follow for the best output. So that kind of interaction with Tamal has always been good. So time to time he just shares his own stories which he was inspired by. You listen to them, then you also feel a little bit inspired. And
Mhm.
yeah, so he's very supportive like that. Just approach. And team member wise, many of the discussions actually happen on coffee or tea table, right? You're going for a walk, it's really good weather outside, you start talking. Among other nonsense,
Mhm.
you do talk about a little bit of science. Oh, you found this. Oh, what is happening next? Oh, you found this. So that kind of
Yes.
discussion in a team, I feel is very important. So
Mhm.
you get ideas from other people's work, other people's perspectives. So that helps a lot. So yeah, I think a good support system can help you think better and better about your work.
Mhm.
And yeah, that is what I feel.
And what were your escape mechanisms? Definitely, we all get frustrated while PhD. And so, what was your mechanism to, shut off and
I
switch back to the.
I had couple of directions that I had kept for myself that if I ever get frustrated or bored in lab, I always had some other activity that equally fascinates me outside lab. So even if I'm feeling bad that this experiment didn't work out, what nonsense, then I'll just go out and do the other thing that I like. So over the years, it was some or other kind of activities like badminton or volleyball or cycling. So even if I feel bad about what is happening in lab, I go with a positive mindset. Yo, I'll enjoy playing badminton today. So that was one to deal with day-to-day frustration. But long-term plan, if I
Mhm.
really, really didn't like PhD, so I was like, hey, fuck it all. I'll just buy a microscope and stick to a room in my house and stay in slides, collect samples from everywhere, whatever is outside, and just look at it. So that was the alternate plan, but I don't think I have reached there yet. Let's see. But I always kept some activity I like outside lab. That was the coping mechanism, if
Mhm.
that's what you're looking for. Yeah.
To maintain the work-life balance.
Yeah. So I should not, my source of happiness should not be my work only. I can feel happy from different things. Sometimes work is
Mhm.
not good. It will get better. What can I do? You can only work on it. I
True,
don't
true,
want to feel sad all the time in other things also.
true. And it brings me to the another question, what I usually ask when I talk to women scientists. Um, Although it doesn't appear, but did you face any particular difficulty just because you are a woman in science?
Luckily for me, so far I have not faced any gender-based discrimination in doing science or anything. But I
Hmm.
do note it may not be happening to me, but it is definitely happening to other people. And one thing I have very clearly noticed is the number of female faculties, let's say
Hmm.
in TIF or Hyderabad. The ratios are very unbiased. So there must be some problem somewhere that we are losing out women as we go higher and higher into the academic field. Even I read an
Hmm.
article somewhere that even in grant applications, number of grant awardees
Hmm.
are less female compared to total applications. So that is there. I personally haven't felt any discrimination, but this does exist. Yeah.
Okay. And another question of what I usually ask is I'm talking to someone of they are at the level of a PI or becoming a PI. What kind of changes you would suggest for the institutes, for the PIs, potential PIs, to make the environment, their lab environment and their office environment more nurturing to the women and female scientists? What would you suggest from
Uh,
your experience?
so many of the problems that happen in day-to-day life, I think it's because lack of awareness. Like it's a lot of social conditioning people grow up with, like the
Hmm.
boundaries of, let's say I'm sitting here. I really don't like people if sit very close to me. So I tell them, you don't sit here. Like I make my boundaries very clear. But sometimes
Hmm.
other people can perceive that these are the boundaries we should be maintaining in the workplace, either in physical space wise or the way we talk. So many people are conditioned to ignore those boundaries. So I think, for example, professionally, let's say you have a lab meeting and everyone is suggesting their opinion or giving some ideas. Each opinion is equally valid, irrespective of the gender. So if I, as a PI, feel like some of the women participants in my lab or in my team are not getting enough time to voice their opinion, I'll tell rest of my lab member we'll go one by one, but we'll so professionally I can these are the things that I can do or suggest to improve women participation. And many of the things men just make women uncomfortable around them. I don't know why that exists, but it happens. So beyond professionalism in social setting, people do need to learn about, they need to be aware of, boundaries with different people, either interacting, body language, words you speak.
Hmm.
That kind of training or sensitization needs to happen at much younger age. By the time, let's say, by the time you are 25, many of your ideologies or thinking are like ingrained into yourself. It's very difficult to change. So I think at younger stage, we need to make all genders more aware of each other and what is right, what is wrong. But professionally, those are the things like giving everyone equal opportunity, everyone's opinion is valid, time is valid, like that. Yeah.
Okay, yeah, I think we have a long way to go for that.
We have a long way
And
to
I think
go.
it, yeah, yeah. in that sense, like because I have some of my friends who are working in very very well-known institutes for that matter, and they openly face that gender bias. So I think not only at personal level, at I think at the level where PIs are there, male or woman for that matter, they have to make this kind of environment, and institutional heads as well. I think they need to take some steps to make these things, to make the environment, working environment, more nurturing and comfortable instead of
Yeah, so
just simply letting it through. Yeah.
the current mindset I feel, which is the dominant one at this point, is like if something happens, like you question why it happened from a perspective of the victim or whoever it is happening to, instead of trying to figure out
Mhm.
what were the social cues or environments that led to that happening. We're not trying to solve at core. We're just trying to suppress the symptoms, like ignore them.
Yes,
Yeah.
yes,
So
yes.
that kind of change needs to happen over a very long time, and it will take a lot of effort. I don't think
Mhm.
it's going to
Yes, yes, yes.
happen anytime soon. Yeah, that's that.
Mhm. Yeah, it can be done for sure. It will take time though, and someone must begin, and it should happen. It's not that hopeless situation. I hope that it should change in near future. And
Very
so
less
as
hope.
I hope
But we all
I
live in
hope this
a
hope
little bit of
increases
hope.
with
So
time. Yeah,
yeah,
yeah.
let's see.
There, there, there must be light somewhere at the end of the tunnel. And as we are reaching towards the end of this beautiful conversation, what would be your take-home message to maybe someone who is in Jawahar Navodaya Vidyalaya and looking to be a scientist, or someone who they are in their bachelors and bit confused whether they should go forward with the further PhD or not, and to those who are in their PhD and struggling a bit to find the end of their PhD. So your take-home messages for all these three people, what would be that?
All these three category of people. So for JNB people, I think that's schooling system. I really like that. Like I have a lot of who I am is dependent on the fact that I was in JNB. So I think they
Mm-hmm.
are in good environment. But it's more of a schooling problem than JNB problem that immediately after 12 people are unaware of what are the other options to do other than MBBS and engineering. So I think if they see people, other scientists around in
Mm-hmm.
media somewhere, they should think of doing all that. And maybe people like us can go back to school and tell them that these are other options that exist and you should think of it. So JNV is a good place. People are good at many different things, not just academia. So at that time, whatever you are passionate about, I will suggest just try to follow that. May or may not be scientist, but
Mm-hmm.
if you are looking into getting into scientist, like yeah, like
Mm-hmm.
try to follow that. Try to interact. Try to get positive push from people who can help you, like either interacting with existing like the
Mm-hmm.
alumni scientists or talking to a biology teacher, not biology teacher, but any teacher that can help you with exploring the future beyond MBBS and engineering are fantastic, but there are also other fantastic options. Other fantastic options for BHC MSc
Mm-hmm.
students. Uh, during my PhD, when I read all these different books, cell biology, molecular biology, it was certainly a different world for me in microbiology. So I was really fascinated by it. So, but a lot of people may or may not like that thing. So I think that is a period where I would suggest people to find their interest again. It may or may not be
Mm-hmm.
academia, but if it is academia, then it's you can start with reading old papers, classical papers. Imagine, let's say, doing some experiment if Golgi reorient Golgi something about Golgi. Back in
Mm-hmm.
Camilo Golgi's time, he was just used to stay in sample and see. So we can read those old papers where there's nothing else available. You just do with one thing that is available to you. You just try to make it work. So I would suggest to find
Mm-hmm.
inspiration in those old, really classical papers for PhD people who are going through whatever normal process of routine of PhD, I would say
Mm-hmm.
कि it will finish, you will complete your thesis, your project, you will get your doctorate. Just hold on. It may feel bad, but you there are parts where which you like also, like forever during your PhD you will end up with this feeling that you have discovered something new. Like for me that is very serotonin inducing, so I like that. So whenever I'm feeling bad, I know that I have discovered something new, and it's fine. Bad time, I'll go through it, but everyone will get their PhD, everyone is going to finish their thesis. Just go through it. But if you're really really sad, do not want to stay in science, I would suggest find something that you really like.
Okay, nice. I think those are quite practical. And one for those actually I do, because I do receive such messages. Like students have done their masters and they are struggling to find PhD guide or institute, and some, for example, those who have not they don't have this gate or net, so they are in a bit of a panic situation, or bit of a in a situation like they want to do it but they are not getting, and bit frustrated. And as I am asking, because you were telling you got in TIFR because you didn't get anywhere, and now you, after a few months, you will be in Harvard, related,
Yeah.
connected to the Harvard. So
Yeah.
what would be your message to them?
I don't know how that part happened. I joined TFR because nothing else worked for me. But if TFR also had not worked, I would have still continued applying for PhDs, because I really wanted to do all this, the whole thing, science and
Hmm.
PhD.
Hmm.
So people who are like that, who just want to do science, I think it may not work out this year, but it will work out either in India or somewhere else. You will get a PhD and find which work interests you, which lab you like, and it's in somewhere or the other, like people find their own way of communicating what is of value to them, right? So
Yes, yes.
somebody will appreciate that value, and you will get that PhD. Just it doesn't mean like five institutes said no to you. Doesn't mean that you will not get anywhere. I am doing fine, so I think everyone can do.
Yeah, that's
That's
more than fine. And thank you so much uh, for like sharing all those beautiful instances and stories. Any particular instance you remember during your PhD that if you look back, oh yeah, that was something I will always remember.
uh, yeah, so during my PhD, at one point, I had to show which cytoskeleton is involved in this process, actin or microtubule.
Hmm.
And generally, whenever Golgi work is involved, most people see microtubule is an important feature or factor there.
Yes.
But for mine, it was not microtubule and it was actin. So I was like, yeah,
Hmm.
I'm going against this flow. So yes, that was my, yeah,
And you did find out that it was actin, right?
it was actin, yeah.
Mm. So those who don't know it, these are the motor molecules involved in
Yeah, these are like the
cell movement.
skeleton of the cell. Yeah, so both microtubule and actin are they help the cell move amongst many other things.
Hmm. And people can find YouTube videos. They look so cool in 3D videos, taking
Yes,
things.
yeah. Many of them actually are submitted for this Nikon or Olympus microscopic image competition. So people should look at the Nikon
Oh,
Small World imaging competition. They have really fantastic images, cytoskeleton and otherwise
uh-huh.
also. That could be
Okay.
inspiring to many people, PhD or BSc, MSc or
Uh-huh.
school students.
Uh-huh. Nice. So the last question, anyone want to ask, they can ask it now. And because it's the last question, have anyone asked you, और कितना पढ़ोगे? अब तो बहुत हो गया.
I cannot tell how many people have asked them, and the top of my list is my mom and dad. और कितना पढ़ोगे? And I'm like, कि you guys chill. I will study. I'll tell you when I stop. So yeah. But people keep asking, और कितना पढ़ोगे? And I'm like, जब तक मेरा मन करेगा, मैं तब तक पढ़ूंगी. Done.
जब तक है जान, जब तक है जान, जब तक है
जब
जान.
तक है जान. Yeah. True.
And all that pressure goes to your sibling, as I was reading one of your interview.
Yeah, so since I'm a third child, so I think many of the trial and error methods on them have created an
Hmm.
environment where I actually can do anything I want. So, yeah, I will
Hmm.
keep studying.
Nice. And I really hope that sometime I get to meet you and personally we can talk about
Yeah,
actin and
actually, I
microtubules
was
and
hoping we could interact personally. I really like that you named one of the species after our college. That was fantastic.
yes, yes.
I am following your
Yeah,
work
yeah.
actually. I see all your research articles. Yeah.
Thank
Oh,
you.
we'll be in touch.
Yeah, yeah, sure. And maybe one day I'll find something related to that in some other species of crabs. Something that molecular.
I'm actually really fascinated by almost transparent nature of many of the aquatic animals. And I'm fascinated
Hmm.
what would be happening at intracellular level that they are transparent. Some fishes are transparent, right? Like
Yes,
see through. Like how
yes,
does
yes,
that
yes.
happen?
Yeah. Yeah,
Intracellularly,
yeah.
I'm really fascinated about that.
Oh. Oh, so who knows after 10 years we might have a collaboration.
We might have, yeah. Let's see.
Okay, it was really nice talking to you, Purnati, and I really had a great time. And thank you so much for sharing all the experiences. And that's it.
Yeah. Thank you.
Thank you.
Thank you for having
Thank
me.
you. Thank. Yeah.